Um, Hi everyone. My name's Elisa Clemente. I was a very horrible baby. I spent many, many hours crying as a baby, so I guess it's fair that I now spend many, many hours just to introduce myself as an adult. Unfortunately, this extremely long name did not come with the social status that you'd expect, so here I am today, a scientist. Uh, I say I'm a scientist. I'm not quite a scientist just yet. I'm a PhD student. And PhD students are to actual scientists what medical students are to actual medical doctors. So they're like younger, but more incompetent and just overall worse versions of actual scientists. Or maybe I'm just projecting here. I actually thought about doing medicine at some point, but decided to do a PhD in neuroscience instead. That's because I didn't want to spend many, many, many years of my life learning about lots of things I hated and the few things I loved. So I decided to instead spend many, many, many years of my life learning to hate the few things I loved. Why all the hatred, Elisa? Is it because it has now been 295 days since you've last had an experiment to work? Yeah, it is actually, yes. But I'm feeling really hopeful now, actually, because of the current situation. Many people have been away from the labs. And so now universities and funding bodies have to choose between, you know, fulfilling the incredible dream of, the noble pursuit of knowledge and therefore giving paid extensions to dozens of thousands of PhD students or lowering their standards. And we all know what they're going to choose. So I finally have a chance to pass my PhD now. anyway, a PhD in neuroscience is really, really good because people overestimate your intelligence and it's a lot more glamorous than the actual day-to-day life. I spend about 99% of my day-to-day life handling dead fish. Now, this is not because I can't keep them alive. I have been very successful at keeping quite a few cacti alive over the years, actually. Thank you very much. The reason why I work with dead fish so much is because I work with fish DNA. And as we've heard before already, I need to first kill them and then mash them up. And then I can do my job normally. It's really not that glamorous after all. And I talk about dead fish so much that I'm pretty sure most of my family think that I'm training to be a fishmonger or something like that. At least that would explain the low pay. But anyway, it's okay, because when you're at a bar chatting someone up, the conversation doesn't normally get this far. It never gets to dead fish. It normally ends right at scientist. You just see their eyes, and they're gone immediately. Now, I know I said I work with fish DNA, but that's actually incredibly inaccurate. I work with fish RNA. Who knew one letter could make such a big difference? Now, if you're fortunate enough not to know what RNA is, it's basically DNA's evil fraternal twin. So it's a molecule that's produced from DNA, and scientists like to study it and like to use it because it gives you an indication of which genes are important for specific cells at whatever time point. And that's really, really, really interesting because it's not actually that interesting. But, I've spent 2.5 years on it, so it's a bit too late to go back now, unfortunately. That would have been a waste of time, wouldn't it? Anyway, this protocol is called in situ hybridization. Sexy, no? Okay, maybe it was just me. Anyway, don't let the Latin in the name fool you. This technique is a work of the devil. Maybe it's just for me. Basically, my job is to take these dead fish, use this in situ hybridization, and essentially try to see where specific RNA molecules are present in those fish at that time. So for you to get a small idea of what it's like, RNA is really, really fragile. It's like a PhD student's ego. And it's also
Friends, I think Elisa is
more than
frozen.
my
Oh,
job
wait.
prospects after my PhD. Sorry, how far? Where was I? Okay. So RNA, as I was saying, is really, really small, smaller than an organ, smaller than a cell, smaller even than my job prospects after my PhD. So really, really small. It's a bit like I'm doing Finding Nemo, but on a smaller scale. So I spent about 2.5 years trying to do this, basically crying over dead fish all the time because it never, ever worked. But it turns out that to get this to work, all you had to do was change a single step in a 4-day, 52-step long protocol that I refused to repeat ever again after my PhD. And so now, after all this time, it's finally working. So I'm really excited and it's so relieved and so happy and so proud because I have these beautiful, cute, amazing pictures of dead fish. That sounded much better in my head. Thank you.